I'm Daniel Levine, and this is the Bio Report. There have been great advances in the treatment of hepatitis C with the advent of curative therapies, but hepatitis B has proven far more elusive. That's due to the differences in the way the virus replicates and how it creates a reservoir of viral DNA in the cells of the liver. nChroma Bio, the result of a merger between Chroma Medicine and Nvelop Therapeutics, thinks it has an answer. It's developing a one-and-done epigenetic editing therapy that silences hepatitis B viral transcription. We spoke to nChroma Bio Chief Development Officer Jenny Marlowe and Chief Scientific Officer Melissa Bonner about its experimental epigenetic editor for hepatitis B, the merger that brought together the two companies, and how it plans to leverage Chroma's epigenetic editing platform with Nvelop's programmable, non-viral delivery technologies in future therapeutics. Jenny, Melissa, thanks for joining us.
Absolutely. Thank you for having us.
Yeah, thanks for having us.
We're going to talk about hepatitis B, nChroma Bio, and its efforts to develop a gene therapy to address this. Let's start with hepatitis B, though. For listeners not familiar with the condition, what is it?
Sure. Yeah, I can give an overview of the disease, and then maybe we'll get into a couple other aspects as well. So hepatitis B is a liver disease. It's caused by infection of the hepatitis B virus, also called HBV. I'll probably say HBV a lot through this discussion. Infections with this virus, they really range from something that can be relatively mild and short term. This is called acute hepatitis B. Whereas what we are really trying to address is the more serious long-term infection associated with hepatitis B, which is called chronic hepatitis B. Chronic hepatitis B is characterized by inflammation and swelling of the liver and can actually result in more severe effects as well, such as cirrhosis and liver cancer over the lifetime of the patient. There are about 300 million people globally that live with chronic hepatitis B. And because there's no curative therapies available at the moment, there really is a pretty significant unmet need for patients.
How do people generally contract the disease?
Right. So hepatitis B and the virus itself passes from person to person in a similar way to HIV. It passes through blood, semen, other bodily fluids. It does not spread through casual contact like sneezing and coughing. So common mechanisms of infection are things such as sexual contact, needle sharing, accidental needle sticks, and in fact, transmission from mother to infant during childbirth is a significant way in which people actually contract hepatitis B virus.
Is there a predictable progression of the condition?
So there isn't. And as I said, the vast majority of people who actually contract hepatitis B virus actually go through the acute infection only and naturally clear the virus. Once an infection becomes essentially in that chronic phase, which is more than 6 months of time, that is the point at which patients start to become at risk for those other more serious manifestations that I referenced earlier. But there's no predictability as far as who will clear the virus and who will not.
There are existing therapies that can suppress viral replication or slow the disease progression and perhaps help avoid some of the serious complications like cirrhosis or liver cancer. But why haven't we seen the same type of success that we've seen with hepatitis C, where there have been functional cures?
Right. Yeah. So that's a good question. There's a pretty clear explanation as to why these two viruses are quite different in that regard. So Hepatitis B is definitely harder to cure than hepatitis C. Hepatitis C at this point in time is a highly curable infection. We can go into that as to why. But the main reason for the difference between hepatitis B and C is due to the unique way that the viruses replicate and persist in the body. So hepatitis B virus establishes a reservoir of DNA in liver cells. So there are actually two reservoirs. The primary one is called covalently closed circular DNA, and it's referred to as cccDNA, and I might use that term again during our conversation. That cccDNA sits within infected liver cells and serves as a template for ongoing viral replication. That cccDNA is difficult, if not impossible, to eradicate with antiviral therapies. And this leads to persistent infection, even when other viral markers, so like protein biomarkers of the virus, are suppressed. So antiviral medications do suppress HBV replication, but they don't eliminate that reservoir. That's really the key to why HBV is so difficult to eradicate. And we'll get into that when we start talking about nChroma Bio's approach to what we think is going to be a curative therapy for HBV. So that virus, that viral reservoir, actually then reactivates the moment treatment is stopped when patients are on something like a nucleoside inhibitor. And this is true actually for neuromodalities that are currently in clinical trials as well, such as antisense oligos that act by degrading viral RNA molecules so that viral proteins are not made. While those modalities really have shown great promise in clinical trials with higher functional cure rates effectively than some of the antiviral therapies, it's still relatively low. And again, that's because those therapies are not addressing that direct mechanism of that cccDNA reservoir.
nChroma's lead candidate is a one-and-done gene editing therapy for Hep B, but I'd like you to take a step back and explain how the company came about. This is a result of a merger between Chroma Medicine, which has an epigenetic editing platform, and Nvelop Therapeutics, which is developing programmable non-viral delivery technologies. How did the merger come about, and what was the thinking behind it?
Yeah, that's a great question. So, Chroma Medicine and Nvelop Therapeutics were in pretty unique positions of having great assets on very different ends of the development spectrum. So, Chroma was marching towards the CTA filing for Chroma 1001, which we're going to talk about shortly. And Nvelop had early promising data on targeted in vivo delivery using engineered viral like particles, or EVLPs, which is a term I will likely use in the future. And the two parent companies were essentially looking for each other. So Chroma's best-in-class epigenetic editor cargo was in search of an in vivo delivery modality to expand beyond the liver, and Nvelop's novel modular targeted delivery platform was in need of a cargo. And the merger was a recognition of the strong data that both parent companies had with their complementary platform technologies, effectively bringing together this best-in-class cargo with the epigenetic editing technology and the targeted delivery for extrahepatic tissues with Nvelop's modular EVLP technology. We think that consolidation is a smart path forward, particularly in this economy, and we were fortunate that we had two supportive board of directors and investors who recognized the value of this merger. I think one detail that might not be well understood is, quite frankly, how quickly this merger came together. I think we started talking to each other about 11 weeks before the ink was dry, which I think speaks to how supportive and excited the board and investors were to see these two companies come together. The merger was able to bring together our balance sheets, and we were able to accomplish a capital raise with some top tier investors so that we can continue to prosecute our compelling science for the benefit of patients. And just 6 months post-merger, we're now at the precipice of a true validation for the field of in vivo epigenetic modulation, with many clinical programs expected to release data in the next 6 to 18 months, including our own.
As you say, the merger made sense from a scientific and a product basis, but I'm wondering what role the financing environment played in this, and how the two of you became aware of each other.
Jenny and I, or
No,
Companies.
no, and Nvelop and Chroma.
Seriously.
Yeah, Melissa and I go way
No,
back.
did you have common investors, or did
Yes.
CEOs know each other, or what? How did the discussion begin?
Yeah, I mean, all of the above. I think I think, again, we had some common investors and common board members who really kind of were fortuitously looking to the future and kind of seeing, the state of the state and understanding that this is a really good solution. Again, to bring together these two technologies that are incredibly promising and had great data to basically solve a gap that each other had. This allows us to start thinking about how we can get epigenetic editors beyond the liver to expand into new therapeutic areas. We have really promising data with our EVLP platform in HSPCs. We have really promising data using various of our delivery technologies, obviously in the liver and then in the CNS. So I think this kind of allows us to really think about how can we really understand and push the limits of the technologies that we now have in our toolbox. We have an expanded toolbox.
CRMA 1001, your lead asset was actually originally developed by Chroma Medicine. It's not using the Nvelop technology. What does CRMA 1001 consist of?
Yeah, great question. So Chroma 1001 is an epigenetic editor that employs an mRNA construct and a guide RNA that specifically targets the HBV DNA that Jenny just referenced. That is critically important, those reservoirs for treatment of HBV. They're both formulated in a lipid nanoparticle for delivery by intravenous infusion. So generally, epigenetic editors are fusion proteins that contain a DNA binding domain and a methylation domain. Chroma 1001 specifically methylates the HBV DNA sequences, both in that integrated HBV DNA and in that CCC DNA, which results in the transcriptional silencing of HBV. So delivery of Chroma 1001 to hepatocytes is expected to result in epigenetic silencing of the HBV viral transcripts from both of those sources of DNA. And importantly, epigenetic editors are able to durably alter gene expression of targeted genes without cutting or nicking the DNA. And this is why epigenetic modulation represents a potentially safer and more natural mechanism for silencing target genes via targeted methylation versus gene editing. And yes, it does not incorporate Nvelop's kind of parent technology, but more broadly, nChroma is working with an expanded toolbox following the merger of the two companies, and our future therapies, may consist of RNAs delivered via LNPs or RNPs delivered via EVLPs or other combinations. And as an example, we have a near DC ready PCSK9 asset that utilizes a next generation epigenetic editor that has been demonstrated to be, 3 to 6-fold more potent than competitor editors, including base editors and other epigenetic editors. And we're also exploring approaches to multiplex silencing of several targets at once, including additional cardiovascular targets combined with PCSK9, for example, Lp(a).
So for listeners who may not be familiar with epigenetics, can you explain the epigenome and its role in gene activation and inactivation?
Yes. So sometimes when we think about the genome or a gene, we think about it as almost a linear object. So a gene typically has a start and an end, and whether or not that gene is expressed in a given cell is largely dependent on what is in and around the start sequence of that gene. Now, this is a pretty simplistic view. The genome is actually much more structurally complex and has many layers of control. One of those layers is the epigenome. So the epigenome is essentially chemical modifications to the DNA that control whether or not a gene is expressed. But as these modifications do not impact the actual sequence of the DNA, that linear view is preserved. So, for example, if the DNA on or in close proximity to a gene is chemically modified with a methyl group, that gene will be repressed, and vice versa. Importantly, the epigenome is heritable, which means that these changes are durable and will be passed on to daughter cells.
And as I understand it, you're using the epigenome to silence the HBV DNA at the transcriptional level to inactivate the virus. Can you explain how CRMA 1001 does that?
Sure, yeah. So we've already highlighted the critical importance of CCC DNA in particular, and that is that DNA reservoir of HBV that resides in hepatocytes when patients have chronic hepatitis B. There's also that integrated HBV DNA, and both of these are the reason that cures are so elusive in chronic HBV patients. And this is the most important aspect of nChroma's approach to treating chronic HBV. And also why we believe we have an opportunity to significantly impact the poor functional cure rates that I already mentioned of current treatments like nucleoside inhibitors. And that is the ability to permanently silence transcription from both of those DNA reservoirs of HBV, that cccDNA and the integrated DNA. So as we also talked about previously, Chroma 1001 contains an mRNA, and this mRNA codes for a fusion protein that includes a region that binds to DNA and a region that methylates DNA. This protein is directed by a guide RNA. Melissa already mentioned this. That guide is also contained in the Chroma 1001 drug product, and that guide directs that fusion protein right to the HBV genome. That fusion protein methylates certain nucleotides around that guide RNA target site, and that methylated DNA then is rendered transcriptionally silent. Viral protein expression is suppressed, and as Melissa again already mentioned, those new methylation patterns are permanent in cells. That means that suppression of protein expression is also permanent. So just to extend on that a little bit, our preclinical studies show that Chroma 1001 can silence expression of viral proteins from both forms of HBV DNA. We have two different models that we've used, one which is a model of cccDNA, the other is a model of integrated DNA. Chroma 1001 deposits methylation across both of those forms of DNA and silences transcription from both. This technology, again, also silences transcription without cutting or nicking the DNA, as is the case with other classes of gene editors that are designed also to silence gene expression. We think this is actually especially important in HBV. As in HBV, there are potentially multiple copies of integrated HBV DNA in any given liver cell, which could increase the risk for translocations or chromosomal rearrangements when using other classes of gene editors that actually do directly cut the genome.
And what's known about it from the work you've done to date in terms of its safety and efficacy?
Sure, yeah. So I mentioned a little bit about our preclinical data. I can go into a little more detail on that. But first, let me say that nChroma really is leading the field of epigenetic silencing. We've done years of optimization work on the structure and the design of our epigenetic cargo, resulting in what we see as best-in-class potency, with Chroma 1001 being about 3 times more potent than our competitors. I've already mentioned our preclinical studies that show permanent silencing of protein expression in models of both cccDNA and integrated DNA. These experiments were actually conducted in mice, and we observed silencing in both of these models out to 6 months. That's about as long as we actually ran those studies in mice, and that was after a single dose of Chroma 1001. We've also observed that multiple doses in that animal model, or both of those animal models, results in even higher reductions in protein expression. And in many cases, we've actually seen the complete elimination of viral protein expression in these models, which is actually the ultimate goal that we have in the clinic. We also have really nice data sets in human cell models and in mice directly showing that methylation pattern induced by Chroma 1001 on the HBV DNA and confirms that direct mechanism of action of the product. From a safety perspective, our product looks very similar to other LNP mRNA products that are in preclinical and clinical testing currently. And while our GLP toxicology study is actually still ongoing, we're still working to finalize it. We really expect no major surprises given what we've learned so far about the product, and we'll be finishing up those studies in the coming months.
What's the development path forward?
Yeah, so as I mentioned, we're currently wrapping up those preclinical studies. We're preparing our preclinical and quality packages for submission to regulators. We expect to submit later this year and to begin dose escalation studies in patients with chronic HBV shortly after that. Our discussions over the past year or so really make it clear that HBV experts and treating physicians are really excited to see what epigenetic editors can do for HBV. We have a hopeful and engaged physician and patient community behind us. There's many experts in the field that have actually referred to this new class of genetic medicines as a potential paradigm shift for their patients. So we really couldn't be more excited to realize the potential of this product. And we believe we have a real opportunity to show cures in a patient population that really have never seen that before. It's never been seen in the world of HBV medicines, not to any substantial rate. And this really remains a very large global indication with significant unmet need. And we think we're going to have a real impact with Chroma 1001.
So targeting the liver hasn't been a challenge with genetic medicines, but how does the incorporation of Nvelop's technology expand what's possible in terms of targeting? And how does that inform your thinking around building out the pipeline?
Yeah, and Chroma's in a great position. We have two complementary platform technologies. We're filing the CTA for Chroma 1001 this year, and we're going to be in the clinic shortly thereafter. And we have a research pipeline with many near-term value inflections that could take us down some really exciting paths. So we have a near DC-ready candidate targeting PCSK9 that utilizes a next-generation epigenetic editor that I mentioned previously that has been demonstrated to be about 3 to 6-fold more potent than competitor editors, which includes base editors and other epigenetic editors. The EVLP technology is something that we're really excited about. And to your point, it enables us to go beyond the liver. LNPs are great at getting to the liver. There's not a problem to solve there. Extrahepatic delivery unlocks opportunities for all cargo types, inclusive of epigenetic editing. And having these tools in our toolbox allows us to think about building drug candidates more holistically, what makes sense for a given disease, rather than force fitting a specific technology to make it work. So we can customize our candidates to prioritize the best tool for the job. And our pipeline strategy is meant to facilitate this decision making by really understanding the modularity of our various platform technologies and the differentiation potential to others that are out there.
So have you identified other programs you're pursuing?
We are actively targeting hematopoietic stem and progenitor cells with our EVLP technology with a direct in vivo administration. And we are also exploring some CNS programs as well, but we have not publicly talked about any specific program.
And do these vectors cross the blood-brain barrier, or would you have to infuse them directly into the brain?
So the EVLPs we've been investigating with kind of more direct administration to the CNS compartment, and we're exploring other technologies as well.
You mentioned that with the merger, you were able to do a capital raise. You raised $75 million. That's a pretty good round for what was a preclinical company at the time. What was the discussion with investors like, and why do you think the company was successful at securing funding?
Yeah, we're really fortunate to be in the position that we are in. You know, we have great investors, and they recognize the value of the merger and bringing these two technologies and teams, frankly, together. And as I mentioned previously, we were essentially looking for each other, right? We each had a technology that solved a gap for the other. And I think the fact that we were able to land this merger in approximately 11 weeks, which is my understanding pretty fast, speaks to the excitement that the investors had. So, I think all in all, the conversations were met with a lot of excitement and enthusiasm for the future.
And how far will existing cash take you, and what's the plan for raising additional capital?
Yeah, we're in a great position now with runway to mid 2027. So we don't have any plans for a near term raise. We will continue to reevaluate as we realize our various value inflection points to determine when makes sense. Of course, we're going to be generating valuable clinical data throughout 2026 in a disease which we believe is the perfect use case for the power of epigenetic silencing, one with a large global need, clear and measurable early biomarkers, an opportunity to reach functional cures even in our early trial, and an extremely engaged and excited clinical and patient community, as Jenny noted earlier. And all of this points to a positive outlook for our ability to raise capital via a variety of potential mechanisms as the field and our own data evolve.
And Chroma Bio Chief Development Officer Jenny Marlowe and Chief Scientific Officer Melissa Bonner. Jenny, Melissa, thanks so much for your time today.
Thank
Thank
you.
you.
Appreciate it.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.